When you get that notification on your phone that says software update available,
Mhm.
and it just sits there, taking up space, nagging you for days on end.
Oh, yeah. Usually because nobody wants to wait 20 minutes for their device to just restart and optimize.
Right, exactly. But when you finally give in and click it, this completely invisible package of code downloads in the background, and it completely rewrites how your device
Right.
operates.
It patches security bugs. It improves the battery life.
Yeah, or maybe it changes the entire visual interface.
Yeah.
It is just a tiny, imperceptible packet of information, but it fundamentally alters the physical, tangible performance of the machine.
Which is actually a brilliant mental model for what we are looking at in the biological world today.
It really is. So, welcome to today's deep dive. Today, we're looking at something that scientists literally used to dismiss as cellular dust.
Yeah, they literally thought it was just microscopic garbage bags that a cell was throwing out into the body.
Right. But it turns out, for you listening, this dust is actually the biological equivalent of those software update flash drives. We're talking about exosomes. So, okay, let's unpack this.
And we aren't just going to list off facts today.
No, definitely not. We are tracing a biological message from the moment it is drafted and packaged by a donor cell all the way to its delivery into damaged tissue, where it effectively, overwrites the code of severe trauma.
We're going to look closely at how these vesicles are engineered, how they break into recipient cells, and the massive hurdle of their incredibly short survival time in the bloodstream.
Yeah, and finally, how researchers are bypassing that hurdle through submucosal administration, specifically deploying them to regenerate the vaginal wall.
What's fascinating here is the sheer diversity of the source material we're pulling from for you today. I mean,
Hmm.
we are looking at a stack of incredibly varied research.
Oh, for sure.
We have the Svetlana Pavlova thesis from the Karolinska Institute, which gives us this highly detailed look at the biodistribution and pharmacokinetics of these vesicles.
And we are pairing that with clinical studies from the Mayo Clinic, looking at vaginal mesh repair in porcine models, alongside some really comprehensive reviews on stem cell therapies.
It's a wild intersection of cellular biology and intimate regenerative medicine.
It really is. But to actually grasp how an injection of, dust can heal severe tissue damage, we have to start at the biogenesis.
Right, we have to define what an exosome actually is.
Exactly. So for the listener trying to visualize this, these are ultra small extracellular vesicles. We are talking like 30 to 160 nanometers in diameter.
Incredibly small.
Yeah.
And they're surrounded by a protective lipid bilayer.
Right. And because you, our audience, know your way around a cell, you know that a lipid bilayer is the standard cellular boundary. It protects the internal cargo from the harsh environment of the extracellular space.
The structure is super familiar, but the way they're formed is entirely unique because most vesicles in the body are formed by shedding.
All right. They just pinch off the outer surface of a cell membrane, bud outward, and float away.
Exactly. But exosomes are synthesized through a much more complex internal process called double invagination.
Double invagination. Okay, so an inward folding, but happening twice. I'm trying to picture the mechanics of that. It sounds like a cell, I don't know, swallowing its own membrane.
That is functionally what is happening, yeah.
Yeah.
So first, a section of the outer cell membrane pinches inward, creating a little pocket that seals off into a bubble inside the cell.
Okay, got it.
That structure is called an early endosome. Now, as that endosome matures, the second invagination occurs.
So the membrane of that internal bubble pinches inward again.
Exactly, creating even tinier bubbles inside itself.
Wow, okay, so you end up with a bubble inside the main cell.
Yeah, and at this stage, that larger internal bubble is called a multivesicular body, or MVB. And the tiny bubbles trapped inside it are called intraluminal vesicles, or ILVs.
ILVs, right.
Eventually, that larger MVB navigates back to the outer edge of the cell, fuses with the cell membrane, and just bursts open.
Releasing all those tiny ILVs out into the extracellular space, and only once they're released are they officially classified as exosomes.
That's right.
But the cell isn't just randomly sweeping cytoplasm and like random proteins into these bubbles, right? If these are software updates, there has to be a sorting mechanism that selects the specific code.
Oh, the sorting mechanism is highly sophisticated. The cellular machinery responsible for this is the ESCRT protein complex.
Right, the endosomal sorting complex required for transport.
You got it. And it operates in sequential stages.
Yeah.
So ESCRT0 acts as the initial quality control, recognizing and clustering the specific cargo that needs to be packed.
Okay.
Then ESHP-I and II induce the bud formation, physically deforming the membrane to push it inward.
Ah, I see. And then ESRT3.
ESRT3 acts like a pair of biological scissors. It constricts the neck of the bud and snips the membrane to form the final sealed vesicle.
That is so cool. And the cargo they are packing is, I mean, it's heavy-hitting biological material. It's messenger RNA, microRNA, DNA fragments, specific proteins, lipids.
Very powerful stuff.
But here's the hurdle that always trips me up. Once this exosome is floating around in the extracellular space, how does it get its cargo inside a new target cell?
Well, that's the tricky part.
Yeah, I'm trying to picture the actual physical entry. I mean, they can't just be tiny water balloons bursting against a target cell's exterior, right?
No, definitely not. It requires a highly coordinated cellular endocytosis. The exosome has to be actively taken in by the recipient cell.
Right.
Sometimes this happens via direct membrane fusion, where the lipid bilayer of the exosome simply melts into the target cell's membrane, releasing the contents directly.
Okay.
But often it involves highly specific receptor-ligand interactions.
I always think of that like a Trojan horse scenario. You know, the exosome floats up to the target cell and it possesses the exact molecular keys, like specific surface proteins, to convince the target cell's security gates that it belongs there.
Right, prompting the cell to actively pull it inside. That is one pathway, yeah. But the sources also detail broader, more sweeping methods of endocytosis, such as macropinocytosis and lipid raft-mediated endocytosis.
Okay, let's pause on those terms because they sound pretty dense. Break down macropinocytosis for me.
So macropinocytosis is essentially the cell taking a massive gulp of its surrounding environment.
A massive gulp.
Yeah, the target cell's membrane ruffles up like a wave, crashes back down onto itself, and swallows a large volume of extracellular fluid.
Oh, wow. And it just captures any exosomes floating in that fluid.
Exactly. It's less targeted than the receptor method, but highly effective for absorbing bulk messengers.
That makes sense. And what about the lipid raft-mediated endocytosis?
So lipid rafts are specialized, stiff islands on the cell membrane that are packed densely with cholesterol and sphingolipids. They act like highly organized docking stations.
Okay, docking stations.
Right, and receptors tend to aggregate in these rafts. So when an exosome approaches, it anchors to this dense, organized section of the membrane,
which then folds inward to pull the exosome inside.
You got it. But regardless of the entry method, the functional result is identical. The exosome is endocytosed, and it releases its cargo, like that microRNA, into the cytoplasm.
Fundamentally altering the target cell's gene expression and behavior, which just sounds like a regenerative medicine dream.
It really does.
If you have a damaged tissue, just inject a bunch of these engineered exosomes, let them get endocytosed, and tell the cells to heal themselves.
Right.
But that leads us directly into the massive bottleneck in exosome therapy, right? The pharmacokinetics problem, specifically their clearance half-life.
Yeah, this is the big issue.
So what does this all mean for you, the patient? If these things are so miraculous, why aren't exosome injections replacing traditional drugs everywhere today?
Well, to understand the bottleneck, we have to look closely at the Svetlana Pavlova thesis.
Right, from the Karolinska Institute.
Exactly. When researchers attempted to administer exosomes intravenously, meaning injecting them directly into the systemic bloodstream, they hit a massive wall.
A huge wall.
The exosomes possess an astonishingly short plasma half-life.
Yeah.
I mean, the Pavlova thesis recorded it at less than 2 minutes.
Less than 2 minutes. Wait, so if a doctor injects these incredibly sophisticated biological messengers into your veins, they're completely swept away by your body's immune system in 120 seconds.
Pretty much.
That's insane. It's like unloading a truck full of supplies in a loading zone, and a fleet of tow trucks hauls it all away before you can even open the boxes.
That's a great analogy. The speed of clearance is staggering.
Yeah.
They are neutralized by the mononuclear phagocyte system, or MPS.
Okay.
Within moments of entering the bloodstream, over 90% of the injected exosomes are filtered into the liver and the spleen.
90%.
Yeah, and there, resident macrophages, which act as the body's cellular garbage disposals, recognize them as foreign particulate matter or cellular debris.
And they just consume them.
Exactly.
And the Pavlova thesis found out exactly how they are making this rapid journey to the liver, right? This detail absolutely blew my mind. The hitchhiking discovery.
Oh, yes.
Yeah.
This was brilliant. Using high-resolution imaging flow cytometry, her team made a remarkable observation.
What was it?
Once exosomes enter the turbulence of the bloodstream, They don't just float freely. They bind transiently to circulating platelets and red blood cells.
They literally hitch a ride on the blood cells.
Yeah, they're basically catching a cab to their own funeral.
Catching a cab to their own funeral? That is so wild.
Essentially, yes. The exact mechanism likely involves electrostatic interactions or specific protein binding, but the result is a highly efficient disposal mechanism.
Right, the blood cells carry them straight into the filtration organs.
And the sources also note that size dictates their exact graveyard.
Oh, really?
Yeah, the liver parenchymal cells tend to capture the vesicles that are smaller than 100 nanometers, while the slightly larger ones get trapped in the biological mesh of the spleen.
Which presents a massive engineering problem. I mean, you cannot heal a severe wound or reprogram a diseased organ if your medicine vanishes in 2 minutes.
Right, you just can't.
So scientists had to invent ways to camouflage exosomes from the immune system or figure out a way to deliver them locally so the liver and spleen never even get a chance to filter them out.
And that necessity has driven some incredible bioengineering solutions to evade clearance. One primary strategy is surface modification.
Okay, surface modification.
Yep. Researchers can chemically engineer the exosome's lipid bilayer. For instance, they use a process called pegylation.
Let's drill into pegylation. How does that actually hide a physical object from a macrophage?
Oh, PEG stands for polyethylene glycol. It's a synthetic polymer.
Okay.
When scientists attach long chains of PEG to the surface of the exosome, those chains aggressively attract water molecules from the surrounding blood.
Oh, I see.
This creates a dense microscopic hydration shell around the exosome.
So when a macrophage bumps into it?
It doesn't detect the foreign proteins of the exosome at all. It just feels a layer of water and bounces right off.
That is genius. It renders the exosome virtually invisible to the immune system.
Completely invisible. They also engineer them to display albumin-binding domains, which similarly cloaks them in native blood proteins to prevent that hitchhiking effect on red blood cells.
Okay, so that handles systemic injection. But what if we bypass the bloodstream altogether?
What do you mean?
Let's say you inject these exosomes locally, right into a damaged muscle or mucosal tissue. Even then, you have local fluid turnover.
Right.
Sweat, tears, mucus, interstitial fluid. Won't those local fluids just wash the exosomes away before they can be endocytosed by the target cells?
That's a great point. Local clearance is slower than systemic clearance, but you're right, it is still far too fast for sustained tissue regeneration.
So how do they fix that?
That is why scientists have turned to hydrogel encapsulation. They load the purified exosomes into biodegradable hydrogels made of polymers like chenosin or PEG.
Oh, it makes me think of like slow-release fertilizer for a garden.
Yes, perfect analogy.
Instead of using a high-pressure hose that blasts the soil and washes all the nutrients down the storm drain instantly, you embed these little nutrient pellets into the dirt.
Right, and they just sit in the soil, slowly breaking down, releasing nutrients exactly where the roots need them over a period of weeks.
Exactly.
The mechanics are very similar. The hydrogel acts as a structural matrix, and the sources show that these specialized hydrogels can create a sustained linear release of exosomes for up to 21 days.
21 days. Wow. This effectively solves the clearance problem for localized treatments then.
It really does. The target cells are continuously bathed in these regenerative signals for weeks, allowing for concentrated localized endocytosis without the need for constant re-injections.
Now, here's where it gets really interesting for you listening. We've mapped out the biogenesis, the endocytosis pathways, the brutal 2-minute systemic half-life, and the hydrogel delivery solutions.
Right?
Now, we apply all of that pharmacology to a groundbreaking clinical focus, intimate regenerative medicine, specifically the submucosal administration of exosomes into the vaginal wall.
This is an area where regenerative medicine is offering a completely new paradigm.
It really is.
Because for conditions like postpartum laxity or postmenopausal atrophy, where a steep decline in estrogen leads to thinning epithelial tissues, severe dryness, loss of elasticity, and chronic discomfort.
Yeah.
Traditional treatments have often been limited to systemic hormone replacement therapy or invasive surgical tightening.
Enter exofemale therapy. This utilizes adipose-derived mesenchymal stem cell exosomes.
Yes, ADMSCs.
Put simply, exosomes derive from fat stem cells, which are renowned for their regenerative properties.
Exactly.
By utilizing localized topical application or injectable administration directly into the submucosal layer of the vaginal tissue, these exosomes completely bypass the systemic bloodstream.
Which is key. The liver and spleen never even see them.
Right. And by embedding them directly into the local tissue matrix, matrix, they promote continuous cellular endocytosis right into the vaginal epithelium and the underlying connective tissue.
And once those local cells pull the exosomes inside, the cargo gets to work. A primary mechanism here is the transfer of proangiogenic factors.
Specifically VEGF, right, vascular endothelial growth factor.
Yes. And VEGF is basically the master signal for angiogenesis.
It instructs the endothelial cells to literally sprout new blood vessels.
Correct. VEGF binds to receptors on the existing local blood vessels, triggering a signaling cascade that forces them to branch out and form new capillary networks.
Which dramatically increases blood flow, oxygenation, and nutrient delivery to the vaginal wall.
Exactly. And simultaneously, the exosomes stimulate resident fibroblasts.
And fibroblasts are the cellular factories responsible for producing collagen and elastin fibers, right?
You got it, which restores the structural integrity and elasticity of the tissue.
So they're essentially treating the root cellular cause of the tissue degradation, rewriting the local environment, rather than just masking the symptoms with a temporary lubricant. But what happens when the damage isn't just natural atrophy? What happens when the tissue damage is severe, like a surgical complication?
Well, that's where things get really fascinating. Our sources include a study from the Mayo Clinic by Kisby and colleagues that specifically looked at vaginal mesh exposure.
Right. And the Kisby study provides a perfect real-world stress test for everything we've discussed today.
It really does. So synthetic vaginal mesh is often used surgically to treat pelvic organ prolapse. However, a major debilitating complication is mesh exposure.
Right. This occurs when the vaginal tissue breaks down and fails to heal over the synthetic mesh, leaving the plastic material exposed in the vaginal canal.
And it is notoriously difficult to treat because blood vessels struggle to bridge across a synthetic barrier. Standard practice is for surgeons to just try and stitch the surrounding tissue closed over the mesh again.
Which sounds painful. So to see if exosomes could overcome this barrier, the Mayo Clinic researchers utilized a porcine model.
Right, pigs.
They surgically implanted 16 polypropylene meshes into the vaginas of pigs. After allowing a week for the tissue to break down and expose the mesh, they attempted different interventions.
And for the control group, they relied on traditional surgical closure, just physically stitching the wound back together.
And as expected, the surgery alone failed. The mechanical tension and the lack of vascularization caused the tissue to break down again, and the mesh re-exposed.
Right, the body simply could not generate new tissue fast enough to heal over a foreign synthetic material.
But then they tested the exosomes. They injected purified exosomes directly into the vaginal tissue surrounding the exposed mesh.
And the physical results were staggering.
They really were. The exosome treatment resulted in partial to full resolution of mesh exposures measuring up to 3 by 3 centimeters in size.
3 by 3 centimeters, that's huge. And the histological data, the microscopic tissue analysis, is what proves the underlying mechanism.
Right. The wound didn't just contract and pull closed.
Exactly. The tissue actually regenerated from the ground up. The exosome-treated tissues featured significantly thicker, regenerated epithelial layers.
How thick are we talking?
We're looking at an average of 208 micrometers thick with the exosome therapy compared to a fragile 80 micrometers for the surgery-only group.
Wow, that is nearly triple the thickness of fresh, new, structurally sound skin. I read that they actually tracked the cellular regeneration using a chemical marker called EDU.
Yes, which incorporates into the DNA of newly dividing cells.
By tracking that marker, they confirmed that the tissue was building de novo, meaning from scratch, throughout the entire epithelium.
Right, the exosomes were literally instructing the local cells to manufacture highly vascularized, thick tissue right over the top of a synthetic plastic mesh.
If we connected this to the bigger picture, the Mayo study validates our entire discussion regarding pharmacokinetics and clearance half-life.
It really does. The researchers knew that even with a local submucosal injection, fluid turnover in the vaginal tract would eventually clear the exosomes.
Right, just like we talked about. So they designed the study to test repeat dosing. They compared a single acute injection against a regimen of weekly injections.
Asking whether sustained presence actually alters the biological outcome.
Precisely the point.
And the repeat dosing regimen yielded significantly greater epithelial proliferation and vastly higher capillary density than the single injection.
It proves that to get the biological message across and fundamentally alter tissue behavior, you must combat the body's natural clearance mechanisms.
Exactly. You either need the slow-release hydrogel matrix we discussed earlier, or you need repeated administrations to outpace local clearance.
You have to keep delivering the software update until the tissue fully installs it.
Such a great way to put it.
It's an incredible full circle moment. I mean, we started this deep dive looking at what scientists used to throw away as cellular dust. But when you look at the mechanics, it is a masterclass in biological engineering.
It really is.
So let's recap the journey for you. Exosomes are these highly sophisticated, nanometer-sized messengers packed with genetic code and synthesized through the complex double invagination of the ESCRT pathway.
And they utilize elegant mechanisms like lipid raft-mediated endocytosis to break into target cells.
Right.
And while their systemic half-life is a brutal 2 to 4 minutes, owing to rapid macrophage clearance in the liver and spleen after hitchhiking on red blood cells, well, we are learning exactly how to outsmart that system.
Through innovations in surface cloaking, slow-release hydrogels, and highly specific submucosal injections into areas like the vaginal wall, researchers are proving that these tiny vesicles can do what traditional drugs cannot.
Exactly.
They can literally rebuild damaged tissues, trigger the sprouting of new blood vessels via VEGF, and restore intimate health at a fundamental cellular level.
This raises an important question and perhaps a profound implication for the future of medicine as a whole. Yeah. If these exosomes perfectly mirror the regenerative state of their donor stem cells, and we possess the engineering capabilities to camouflage them from immediate immune clearance or, deliver them locally to ensure sustained endocytosis.
Where does that leave traditional treatment?
Exactly.
Yeah.
Are we approaching an era where we no longer need synthetic chemical drugs or physical scalpels to heal severe internal wounds?
Oh.
Could a simple, localized injection of engineered vesicles one day reprogram your own mucosal tissues to continuously self-repair, rendering traditional invasive surgery entirely obsolete?
Imagine that reality, receiving a targeted software update for your body that permanently fixes the hardware without a surgeon ever needing to make a single incision.
It's incredible.
Something to think about the next time you see that update notification lingering on your phone screen. Until next time, keep diving deep.